The Broad Discovery Center is a public educational space that showcases the groundbreaking biomedical research happening at the Broad Institute, our partner institutions, and our collaborator sites worldwide. Through our interactive and informative exhibits, you'll learn some of the stories behind how researchers at institutions in Kendall Square and across the globe are working together to better understand and treat all different kinds of disease. Building the Broad Discovery Center has truly been a community effort. Our team of scientists, administrators, consultants, community members, and educators have all come together to build a special place for anyone to engage with cutting-edge biomedical science. Whether you have a background in science or not, all are welcome at the Broad Discovery Center. If you're looking to discover more about Boston-based innovation, we encourage you to stop by. Our interactive learning exhibits offer a deeper look at the complex biology associated with cancer, psychiatric research, infectious disease, rare disease, and other conditions. You can also walk through exhibits to learn more about the journeys, stories, and motivations that drive Broad scientists to pursue disease research. We are so excited to bring this space to you, and we hope to see you at the Broad Discovery Center very soon.
And speaking of Broad and Discovery, good evening, everybody, and welcome to the inaugural talk of the Broad Discovery series. There we go, he gets it. My name is Tom Ulrich. I'm the Associate Director for Science Communications here at the Broad Institute, and it is my true pleasure to welcome you here this evening for this first talk of the new series. Many of you are hopefully familiar with the Science for All Seasons talks that the Institute held for several years. With the launch of the new Broad Discovery Center, which was just featured in the video you just saw, and which those of you in the room also basically saw as you walked into the building this evening, we figured we had an opportunity for a new name and for a new look. So with the Broad Discovery series, we aim to explore the genetic and biological roots of human health and disease, the mechanisms that govern how our cells and our bodies function, new technologies that are changing how science is done, and new progress being made to translate findings into treatments for both common and rare disease. We're excited to be launching this series tonight, and we're also really excited to have you here and joining us both in person and online. I'm going to be joined on stage in a couple of minutes by Paul Blainey and Alex Shalek. Paul is a core institute member of the Broad and is also an associate professor of biological engineering at MIT. Alex is an institute member of Broad. This is a long list here. An institute member of the Broad, a core member of MIT's Institute for Medical Engineering and Science, an associate professor of chemistry at MIT, and he also holds memberships at both the Reagan Institute of MGH, MIT, and Harvard, and at the Koch Institute for Integrative Research at MIT. Paul, you need more titles. You got to balance that out. Paul and Alex both bring an engineering approach and an engineering mindset to their work. And what they're going to be talking about tonight is how working and doing research at that balance point between biology and engineering can open up new ways of asking questions and of exploring biology in ways that would not otherwise be possible. If you have any questions for Paul and Alex after the talk, we encourage you to come down and use the microphones at the front of the room during the Q&A portion of the evening. And if you're watching online, we will be monitoring YouTube chat. And so please, we encourage you to put your questions in there, and we'll do our best to bring you into the discussion. For those of you in the room, we invite you to join us for a reception after the talk in the newly opened Broad Discovery Center, just outside the doors here. And we encourage you to explore the museum's exhibits and interactive elements. And lastly, if you're on social media and you're tweeting or talking about the talk tonight, we encourage you to use the hashtag Broad Discovery Series. And I'm going to bring Paul on stage to kick this off.
All right, well, thanks so much, Tom. It's a pleasure to be here speaking with you, and thanks to everyone in the room and online for your interest and attending tonight. So we're going to start just with a few disclosures about some of the outside engagements that Alex and I have. And we decided to start with this both to ensure the integrity of science, but also to point out how some of the work that we do gets translated out into the real world. And some of that occurs through our connections with industry. So we wanted to acknowledge that. Okay, so we're here to talk about discovery. And in the widest frame, it's a really exciting time to think about discovery in the life sciences. And so thinking beyond the Broad for the scientific enterprise and society as a whole, there's a real interest in discovering not just the fundamentals of life, the basics understanding of what creates and sustains life, but also how human interventions in living systems are going to affect our health, sustainability of industry, that includes agriculture, and how we're going to continue to support the natural world. And that's really, as I'll explain, the engineering side of things. And so I want to talk today about how science and engineering work together generally, and particularly here at the Broad. And this is a focus of my research and Alex's, and we're going to give you a concrete understanding of how that plays out in some of our own work. But I want to start by breaking down these two elements. Discovering the fundamentals of life, that's the science of biology, and intervening in living systems, that's engineering as applied to the life sciences. So let's take biology first. And I'm also want to speak particularly on this slide to students that may be listening and imagining careers in related areas. So what do biologists do? Biologists often spend their time doing experiments in the lab or maybe at a computer. And, oh, is that me? I'm going to get a little help from the back in that. I'm going to keep talking about biologists. And one of the things biologists do as scientists is apply the scientific method. And so that has to do with defining scientific hypotheses, which are a conjecture about a way a system works. In the life sciences, we'd be talking about living systems. And then designing and carrying out experiments to collect data or other types of evidence that are going to either support a certain hypothesis, refute a given hypothesis, or maybe even give us some ideas about new ways of thinking and new hypotheses about the systems we're focused on. Okay, so that's the science of biology, and it's about this fundamental level of understanding how the natural world works. Turning to engineering. Engineering is, by definition, oriented towards applications that are human designed. So engineering is towards a human purpose. And engineers day to day build and work with hardware and software systems that are designed to achieve those application goals. With respect to the life sciences, oftentimes this hardware might itself consist of living systems, organisms, cells, or molecules. Engineers also take a systematic approach in the practice of their work. And so this often encompasses designing and constructing systems, instrumenting those so that we can measure how they operate and their characteristics over time, mining the resulting data to try to understand more about how those design systems work, modeling, creating computer models often of the functioning of those systems so that we can quickly think about what kinds of changes we could make to improve their function, and finally, manipulating the real systems to realize those improved functions. And so engineering is really intrinsic to the life sciences today, and you'll see lots of examples of that through the talk. But I want to emphasize there are a couple different ways that this interface plays out. One is what I mentioned, where engineers may actually design and manipulate biological systems themselves. We've got some great examples of that in the research at the Broad Institute these days. Engineering also impacts biological science and discovery in the life sciences through the design of often instrument-based systems that are used to acquire data that biological scientists want to use in their research. And we'll have lots of examples of that through the talk today as well. All right, biology and engineering together at the Broad. So the state of development of life science research today, we often require acquisition of really large data sets to progress the science. And so this is one of the reasons why engineering is coming to the fore in the life sciences research, because the design of increasingly powerful tools for large-scale data acquisition is helping drive a lot of the science that's at the forefront today. And Broad's been a leader in doing this for many years now. Looking back, there are a couple examples I'll call out that you may have heard about in the popular press or reading scientific articles. One would be DNA sequencing technology. So really developed over the course of the Human Genome Project, and Broad was a partner with a lot of the companies that were developing the leading instruments. This went on to be refined to the extent where we could sequence many, many, many human genomes and use even those whole genome data in genome-wide association studies. Another example would be genome editing. So this includes CRISPR technology. And this is an example where actual biomolecules are engineered to a purpose, in this case, making really precision changes in genomes for research and maybe someday therapeutic purposes. Final example, microfluidics. Microfluidics has to do with manipulating liquids on incredibly small scales precisely and in an automated fashion. And this has supported high-throughput single-cell analysis. We're going to hear more about that from Alex, as well as studies of the microbiome and infectious disease diagnostics that I'll touch on. Okay, so what's next going forward in life science research at this interface of biological science and engineering? Well, the state of the science in many areas now is that we've got a pretty good list of the parts. So that is, what are the cells that make up an organism? What sorts of biomolecules are present in cells? There's more discovery to do there. But a big part of the challenge today is understanding what it is that these parts do, how those parts work together, and how each of those parts contributes to the function of the whole cell or the whole organism. And you can imagine how that's important to human disease. So one of the approaches that's really taking flight today are approaches that perturb the function of these parts composing the systems. And this is a classic approach in engineering. If you've heard the term reverse engineering, you can imagine what this entails. And so, I think of my small children at home who might investigate the function of a toy truck by, for example, I don't know, breaking off all the wheels and then discovering the relationship of those wheels, those parts, to the function of the whole, right? So the wheels contribute to a rolling function of the truck, which is important for some of the engineering objectives that the truck was designed for. All right, so that's the introduction. I'm going to hand it off now to Alex, and I'll come back and see you in a few minutes.
Thanks, Paul. And thanks, everybody, for joining us today, and thanks for having me. It's a real pleasure to be here. And I want to thank Tom for removing a couple of my affiliations to make Paul feel more comfortable in this audience. So it's really cool to be here and to be talking more broadly about what we're trying to do. And Paul introduced this idea that we're at this point now where we can really do an incredible job of measuring the components in biology. And what I was hoping to do was to tell you a little bit about how some of the technologies that we develop can be used to actually figure out those components. But where I wanted to start is with what is the broad thing that our lab is interested in? And that's something that ties in very nicely to what Paul was talking about, like where the big untold challenges are. And the thing that our lab is interested in understanding is what constitutes balance or homeostasis in our tissues. And so this idea of equilibrium or balance is a really simple concept. If you're thinking about weights on a balance, or you're thinking about, for example, in a chemical reaction, I teach this to undergraduates at MIT in 560, under a particular condition, what is equilibrium? It's really simple. It's mathematical. I can derive it. But if somebody was to ask me in our tissues, what is balance? What does that mean at a cellular and molecular level? It'd be really hard to figure out. But I know how important it is. If I have, for example, too much immune activity in an autoimmunity, I can get tissue damage and death. And on the other hand, if I dial down immune activity so I have too little, I can become susceptible to opportunistic infections and the outgrowth of tumors. So what our lab really likes to try and study is what defines balance. What does this homeostasis actually mean? What are the deviations that occur in disease or under different perturbations? And how can we rebalance the system to restore human health or develop vaccines that will help buffer it in the first place? And to do this, we spend a lot of our time developing and applying tools that let us dissect these really complicated multicellular ecosystems that are our tissues. And I'm going to talk about one of them today that I'm going to show you in the corner here, and we'll pass around some examples in a little bit. So stay tuned for that. So what this really means, not surprisingly, because I was trained originally as a chemist and a physicist, I'm really interested in sort of the elements of our body. I'm interested in what are the different cell types that exist, what are their diversity, what are the qualities they have, what is the breadth of their immune cell, what do they target, how durable are they, where do they exist? And right now is an incredible time to think about cellular biology. And the reason for that is that over the past 5 to 10 years, there have been intersecting advances in cellular and molecular biology and also micro-nanotechnology that let us study the properties of cells at unprecedented resolution. So rather than just picking 1 or 2 proteins that we think are important, we can now sequence the genome of an individual cell. We don't have to look at it in aggregate for every single cell. Just like for every individual, we can look at the genome. We can measure what parts are open, which parts can be read by the cell, can be used as instructions for performing operations. We can measure multiple proteins. We can even look at the transcriptome, all the RNA, everything a cell's thinking at a given moment. And so this is fantastic. You might say, well, how does this really work? Well, all we have to do is we have to take our tissues, break them apart into the individual components, just like I might separate each and every one of you out, and then isolate those individual cells, say in one of the small little tubes that you might often see a scientist in some clip art holding. So something that is about 1 milliliter, that's roughly about a 25th of a tablespoon if you need a volume to think about. And within there, we can do some very simple molecular work. So all of our messenger RNA, all of the things that cells are trying to do at a given moment, have this poly A tail that I'm showing here with a bunch of A's. We can use one piece of DNA, this oligo DT primer that's the stretch of T's, to reverse transcribe all the messenger RNA, and then throw it on a sequencer to figure out what a cell is thinking at a given moment. And that lets us think about these cells in this really high dimensional space. So I don't have to say a cell is expressing this gene or that gene. I can say, what are all the genes it's expressing? Here I show you as a point in 3-dimensional space across 3 genes. We're measuring about 20 to 30,000 different points. And so that's really hard to visualize. So very often what we do is we simplify things down like this into these 2-dimensional plots, where each point is a cell. And now what I've done is I basically have grouped the cells in this very high dimensional space and colored them by similarity. From looking at what defines green relative to yellow in a computer, I can figure out what the cell types are, because I can say to myself, well, the green cells express TCR, the T cell receptor, which only shows up on the T cells. So in a way, this is a nice way of discovering biology from first principles. Now, just as an analogy, because I think it's really helpful when you think about what we're doing here, we're measuring all of the RNAs and we're looking for patterns in them. That would be like going to the United States and instead of taking an average education or an average income, going and looking at all of those demographics across the entire United States, like advanced degree, median income, commute time if you want to pick something random, and households rented. You can find when you look that there are certain parameters that associate in a region. So for example, percent advanced degree and median income, and you'll see, as you would expect, that the average weekly income increases with education. And so the idea is the same here. We're going to look at all the genes in the cells. We're going to look at which ones are used together, because that tells us something about associations within cells, within those programs, sort of the fundamental workings of these biological machines. And so you might say, well, how do we do this? Like if you wanted to really understand the cells and types in our body, where would you go? Well, if you're curious, if you were to Google it, maybe you already did on your phone, the number of cells that exist in our body is about 30 trillion. I, for truth in advertising, haven't taken biology since I was in high school. And so I went back to my high school textbook, which I still have thanks to my mother, who doesn't throw anything away. And it tells me that there are 300 major cell types. But if you go pick up an article that's in the public literature in one of the journals, you might see that somebody claims that there are 100 different subtypes of neurons just in the retina alone. So you might say to yourself, well, what really is there? Like, what do we need to know? Well, the first thing you might want to know are what are all the cell types that exist in your body? We know that these types can exist in different states, so you might want to know that. Like an immune cell can be activated where it's trying to fight off a pathogen, or it can be at rest. We might want to understand where they are in your body, because we know that it's important that you have specific cells in your brain and not in other locations. We might want to understand how they relate to one another and can transition, and developmentally how they show up, so that if there's a deviation that we understand where that's coming from. And then finally, and most importantly for some of the work that we do that impacts people more broadly, we want to understand what changes in disease, because when we understand what those differences are, we can think about how to repair them therapeutically. And the problem is that our knowledge is only partial or decades old. So the other thing I'd say, just since we're at the Broad, and this is something that Paul brought up, I think of cells as really important, because cells are sort of machines. They're operating on the information that's in your DNA. And so when you have a mutation that shows up, we need to understand what cell type is using that DNA and how to understand why the particular variant manifests in disease. So it's important to understand what's happening to neurons, for example, if we have a mutation associated with autism, and so on and so forth. So not surprisingly, we're one of many labs that are very interested in creating this reference that would be transformative for the community. So there's an entire group of individuals in this grassroots movement called the Human Cell Atlas. The Broad actually played a large part in helping to found through Aviv Regev, who was here previously, to create this sort of periodic table of all the cells in our body across different tissues, across different cell types, across different modalities. This is really comprehensive reference book, this atlas that you could go to when you wanted to understand, diagnose, monitor, or treat disease. And so you might say, well, that's great. You're at the Broad, you're an engineer, you know how to sequence cells. Why don't we get started? Well, I showed you that little tube that I had in my little diagram, and if you hold it up in your hand and you say, well, all I have to do now is I have to take one cell and put it in the tube, it's great. But there's a fundamental problem, and that's that if I take it and I think about it, we have a size issue. And so I don't like the SATs. I have not very fond memories of them. Maybe all of you love them, and I couldn't exactly remember like the analogy format, but I looked it up online and I found it. And so if we say the cell is to this little tube that is just about the size of your hand where it can slip through because it's so small, you might say, well, if I was to scale that relative to a person, how big would it be? Well, if I gave you that the unit of measure was a football field, would you think it's 1 square football field, 5 square football fields? Yep, I see some thumbs going up in the audience. If you do the math, it actually turns out that it is 10 football fields by 10 football fields by 10 football fields. Pick your favorite sport pitch. It's basically the same thing. And I was going to pick hockey rinks, but I didn't think people would know how big those were. So you can imagine that this is a very luxurious accommodation for a cell. But if you start thinking about how much volume of liquid you're going to need to start looking at even 1,000 cells, maybe even 1 trillion, it's going to be prohibitive. So this is where it becomes important to think about sizes in biology. And so I stole this video from the web. I think it's great. It's going to run on loop, but it helps you to think about how big things are. And as somebody who was never trained in biology, I'll tell you the first time I was talking about proteins relative to DNA, I was like, proteins are the products of DNA. They're probably pretty big. And then somebody was like, no, Alex, they're really small. Like the DNA encodes everything. It's like 2 meters and like gets folded up. And so as you look at it, you can see our cells are about 30 microns, maybe down to 10 microns. You keep on going down and you think about the molecules that we're actually trying to go after. And they're down here in the nanometer length scale, maybe a little bit bigger. And so it's really important to understand what is an appropriate size, what's a spacious living accommodation for a cell. And that's where the micro and nanotechnology comes in. So I was showing you the idea of having like a tube. And so probably you go into a lab and you'd be like, oh, we can do high throughput. Let's take a 96 well plate or a 384 well plate. And that's what I'm showing you here. It's basically 8 by 12 or 16 by 24. And if you look at it, it's got a well that's like 3 millimeters by 3 millimeters. It's got a volume of about 10 microliters, which sounds much smaller than milliliters. So it feels like it's going to be big. But the throughput, as you can see, is sort of about maybe 1 to 100. So you might say to yourself, well, if I don't like using this, maybe I could use advanced technology to create increasingly small devices. And so this is something you can do. And when I was working with some people here in partnership with a company called Fluidigm out in California, we started to use microfluidic devices as a way of isolating cells. And so what you're looking at here, and let me see if I can use the pointer, which doesn't work. So I'm going to use my hands as a shadow puppet, which I was told I was not supposed to do, but here you go for everybody on the screen. Paul, I know you want to do this, and now you have appropriate actions. There's a little cell, and what I want you to see is that this is a little microfluidic channel. It's kind of like a computer chip, where instead of electrons flowing, we're going to have cells in solution. Cell comes in right here. It gets captured in this little cup, which is right there. And then the remainder bypasses around here and goes through this channel. And this looks great when you're looking at it because you see the cell and you think it's really small. But the problem is that these devices work just like your computer chips. They basically store, but then they flow through. So it might be microns in this dimension right here where we can see the cell, but we're still centimeters in this dimension because we have these reagent buses that are passing things along. And what that means is that we get 1,000 times smaller, but we only get a small boost in the number of things that we can look at. And so you might say to yourself, well, I really like that spot where you're capturing the cell. I just don't like everything else. Could you get rid of it? And the answer is yes. You can turn to increasingly simple devices by streamlining and simplifying the processing, like these picowells that I show you here. And these picowells are basically about the width, height, and length of your hair, so about 50 microns by 50 microns by 50 microns. And what that does is it reduces us down now to picoliters. So many orders of reduction now. We started at that milliliter, then we went down 1,000 to microliters, then 1,000 to nanoliters, then 1,000 to picoliters. So we're very small right now. And what that means is that you can actually think about capturing and processing a lot of small cells in a very small volume. And so just to drive that home, if you go and you do the math, if you say a picowell has about 100 picoliters in it per cell, you could now say if I was to take 3 or 30 trillion cells and put them in and do the math, what I'll get at the end of the day is that the volume that I need is roughly 1.5 meters by 1.5 meters by 1.5 meters. And I was glad the math worked out because when I was doing it on a piece of paper, I was like, it should be roughly the size of a person. And it is. It's a slightly big person. But you can imagine now that sounds like a lot of expensive reagent, but it would be possible to start doing things in this way. So with some partners over at the Koch, Chris Love, Todd, Mark, and Travis tried to devise a system for doing single-cell RNA sequencing, for doing this molecular barcoding and processing that I talked about before, using a very simple device. And so I have a couple examples that I'm going to ask Tom to pass around so you can play with them. They're on glass slides, so the edges are sharp. I put a little tape on. Just please try and be careful with your fingers. But the basic idea is that what we're going to do is we're going to isolate individual cells in these little wells right here with some sort of bead that can capture all their molecular information. And what we'll be working with, as you'll see on the slide, if I go over here, are these 1x3 glass slides that are kind of like ice cube trays that really had been pioneered by Chris Love's lab. And so imagine it's got about 100,000 little boxes on it, which are going to be little holding chambers that we can use to capture and interrogate cells. And so what's so nice about this, as you'll see as it comes around, is it's just like this really simple device. You can just put cells on top. And what that means is that you don't need a lot of material. Yeah, we'll be happy to have a lot of material, but we can work with just about anything. So you can take peripheral blood, for example, as I'm showing you on the far left, and try and look at what happens in acute HIV infection to try and understand what's associated with early damage due to the virus, as Sam did in his onesie up top. You can think about things like pinch biopsies that are half the size of a poppy seed to understand what's different between a region of the gut that's inflamed or uninflamed in somebody that might have IBD, as Jose did. And he told me that I owe him royalties for the use of his hand because that picture has shown up many times. And then finally, you can take fluids where you're not even sure that there are cells, like cerebrospinal fluid, and look before and after treatment, say in immunotherapy, to see what's different between individuals that respond or don't respond. And you can see at the top that was work done by Jay, who's also in a onesie. So the big lesson to you is if you join our lab, you should not take a photo in a onesie because it will be used in a presentation that will go on YouTube and haunt you forever. And just to give you a sense of like how low you can go on these things, most recently during the COVID pandemic, we've been using it as a way of characterizing what's happening in mild and severe infection. So those same nasal swabs that you probably have had tickle your brain that you probably hate, rather than using that just to see if you have virus, we can actually take the cells that slough off, load them onto these devices, characterize all the cells in your nose, figure out which ones are infected, and what's different between people with mild or severe disease. So maybe in high school biology, you had to do the same experiment I did, where you took a toothpick, you scraped your cheek, and you looked at it under a microscope. Same thing now, just with genomics. So I wanted to put it all together to show you how this works and what this enables and why Paul said at the beginning, we're at a pretty good place with respect to thinking about the parts list in biology. So what we can do now is we can take a bunch of different tissues and we can isolate out the individual cells by breaking them up into their components using things that are like fancy blenders. We can then isolate those individual cells using a variety of different devices. I talked today about the idea of using tubes or plates or microfluidic circuits. There are lots of other technologies like salad dressings, literally, that you can use to separate cells. And so now you can imagine this array that I'm showing you here that we call sequel for a lot of bad reasons, that we put our cells and our beads into. We then can perform our very simple molecular biology, as I mentioned before. We can go and we can get enough material that we can load it on the sequencer, throw it on our favorite technology, and then we can measure the expression of all 30,000 genes for each individual cell to see what's going on. If we do that, say, for 100,000 cells, what we end up with at the end of the day is this incredible set of data that's about 3 billion data points. And so what I'd say to you is I think there are really exciting opportunities in biology and also in engineering, but I think there's really cool opportunities also in computational biology and in methods to apply data science to the study of this kind of information. So this is what happens if we put all of those cells down and we try and create a social network. So what I've done, and I had to run this in real time because I couldn't click it, I basically have put cells down on the surface, and then I have started to try and pull cells that are similar together and push ones apart, creating a social network. And what you'll see over time as we use way too much computational power here at the Broad and take a lot of snapshots is that cells start to collect together into little groups, some of which are all colored one thing because they come from one tissue, and some of which are more intermixed. So if I stop running the video and I sort of show you what the graphic looks like overall, you get this really beautiful hairball that tells you a lot about what's happening in biology. Now, each point in here is a cell, and what it really is it's a measure of the expression of all 30,000 genes for each cell. By looking at what we see in each, we can identify what we're looking at. So I see some microglia, which are these immune cell types that are associated with defense in the brain, some hepatocytes which are used to clear out potential, environmental contaminants in your liver, and so on and so forth. And you might be asking, well, what's in the middle? It's the immune cells because they're common to all your tissues and a great way of thinking about common denominators. So how do you use this sort of data? Like, why is it so useful? Why is it so powerful? Well, just as a quick example, early in the pandemic, there was a lot of information on what was happening sort of globally in infections, the malaise, the fevers, the symptoms that people get. And on the other hand, people identified that just like with other coronaviruses, that this attached to ACE2 on the surface and used this particular protease, TMPRSS2, and maybe you've heard all about this. What we can do now, because we've measured everything in our cells, is we can go back and ask ourselves, which cells in which tissues are likely to be targets, so we can begin to understand why we're seeing symptoms and start to think about therapies. And so if you go look at some of the data we've collected, you can see that there's high expression of this receptor for the virus in the gut, in the ileum, and in the jejunum. But in other tissues, like say, for example, the spleen, you see very few cells which are points expressing these RNAs. We can use the information we have to resolve down further to figure out what are the real targets that we're looking at. And so here is some lung tissue where we've broken it out into different immune populations like T cells and different epithelial populations, the cells that sort of form the basis of the lung, like type 2 pneumocytes. And as we look in, what we find that's actually really interesting is that there are very few cells that are targetable by the virus, which raises some really interesting questions for why it works in the way in which it does. And so I hope what I've shown you at least here is that the reason that engineering is so important when we think about profiling and measuring things in biology is that we need to scale. If we want to go exponential, the way in which we do it is by shrinking, simplifying, and economizing, which is really what micro and nanotechnology enables us to do in the context of biological discovery. And so with that, I'll turn it back to Paul to talk about all the cool things you can do with little containers.
Great. Thanks, Alex. I'm here to continue the talk with a focus on salad dressing. which I think was Alex's reference to the microdroplet technology. And so what you're seeing here is actually a combination of two different microfluidic paradigms. The metaphorical salad dressing here on the left, combined with this kind of microwell technology that Chris Love's lab and Alex's lab have used to such great effect in single cell analysis. And so what we've been doing is combining those to create a new platform that we call Droplet in Microwell, which you're seeing here with the colored droplets. And I'm going to show you over the course of the next few slides some of the different applications, engineering applications, that we've deployed this new platform for. OK, so before we get into how we're using it now, I want to tell you the origin story of this technology platform and why we decided to ultimately start putting microdroplets, the salad dressing, into the microwells to make a microfluidic masterpiece of sorts. And this went back, 6 or 7 years now, to our interest in doing combinatorial drug screening. So we, at the time, and we still are, very enthusiastic about this idea of doing combination therapy, which is gaining currency in cancer and infectious disease, but we think it has a lot more potential than that. But it requires screening multiple compounds, multiple candidate drug compounds at the same time. And it turns out, even though that sounds simple, it's actually pretty technically hard to do at high throughput. But we think there's really tremendous promise in being able to do this, because the design space for therapeutics expands tremendously if you can use multiple independent compounds to tackle different parts of a biological network. And so we had kind of a general notion that microfluidics might be useful in solving this combinatorial screening problem, something to do with some of the miniaturization and efficiency that Alex was appealing to. And so that's fine, but the plot thickens because there was a problem in the field that just had to be overcome before we could deploy this kind of droplet technology for compound screening. And so you're seeing an illustration of the problem in the movie here. And so you'll see when the movie resets, some of these droplets start off with a dye molecule, which is our stand-in for a drug molecule, and some are empty. But over the time course, you can see that the concentration evens out in each group of 7 droplets here. Yeah? And so what that is that's our metaphorical drug molecule leaking out of some droplets and being taken up by other droplets. And you can imagine that's a terrible system for trying to do a whole bunch of different microscale drug assays with a different pair of drug molecules in each droplet. And so this was the problem I set my lab to solve. So I talked about this problem and the goal of doing the combination drug screening, which is a dream I shared with some of the group members, and sent them off. Okay, go. Think of a way to solve this problem. And I even gave them some really good suggestions, which were promptly dismissed. And sometime later, students came back and they had this beautiful engineering-based solution to the drug leakage problem, and had nothing to do with any of the things I suggested they try. And you're actually also seeing that in the movie here. So the reason the droplets are in these patterns, the hexagons with a drop in the center, is because these groups of 7 droplets are in microwells designed to hold exactly 7 droplets. And what's actually not visible is there's a group of 7 droplets in the middle, which started out by chance with no dye molecule. And what you can see is at the end of the time course, they still don't have dye molecule. So what can we learn from that, right? Well, we can see the 7 droplets here, the 7 droplets you can't see here, failed to pick up any of the dye from all these surrounding droplets that started out filled with dye, right? So we've managed to achieve compartmentalization of the dye molecule, not at the droplet level, where it's leaking all over the place, but at the microwell level, right? So this set of 7 is independent of this set of 7, independent of this set of 7, and so on. So that's the solution. That's why we put the droplets in the microwells, because we can keep the compounds separate at the microwell level. And then if we want to do a combinatorial screen, we can bring compounds into each microwell at the droplet level. In this case, perhaps 7 different compounds coming together. And so this is a system that really hit all our goals for combinatorial compound screening. We can, of course, put cells in these droplets to do cell-based assays and access all those wonderful biological networks that we want to perturb with our drug molecules. The droplets load themselves into the microwells. So isn't that convenient? No person has to go and put them one by one there. No robot has to go put them one by one there. They do it by themselves. And then this is all in this microfluidic world where things are tiny. And even with a small quantity of compound that's easy to access, we can test that given compound in combination with many other compounds without running out of the drug we want to test. So this is an illustration of how we deploy the technology for compound screening. One of the other things we need to do, because all the droplets get mixed up when they go and they're automatically loading themselves into the microwells, is to figure out, okay, once they're loaded, which droplet has which drug in it. And so to do that, we color code them up front, more or less with food coloring, so that under the microscope, we can look at the colors and tell which droplet has which drug in it. And if we could just play the movie one more time. Can I get a little help restarting that? Here it comes. So the movie starts with this big mixed emulsion, this rainbow of droplets getting loaded into the microwell array. And it flows down underneath where the droplets can all pop up and load the microwell array. So that's the droplet charge getting loaded. And you can see it's a very high density array. So we can do a lot of drug assays on one of these devices. Read the color code. And then, of course, we have to fuse those droplets. So we have a way to merge them with an electric field in order to formulate the assays. So you'll see this merge here. There they go. Yeah. So I have one of the arrays here to pass around. Nothing too sharp on it, I think. Yeah, OK. So I want to tell you about a couple of the projects where we've deployed this technology platform. So this is one where we used it to study microbial communities, so groups of microbes growing together. And here we elaborated the platform a little bit, designing microwells that can hold like different specific numbers of droplets. And then you see them here where we've merged the different droplet sets to allow all the droplet inputs to work together. And so the study here was to look at this organism, Herbispirillum, a bacterium, which is a model for plant symbionts. So microbes that help plants grow better. And the question we were asking was, OK, that symbiont, it doesn't just live by itself on plants. If it's out in a farmer's field, it's there with many other different types of microbes. And so we used the combinatorial power of the droplet array platform to test how the growth of that organism was affected by the presence of other combinations of organisms. And we also did this with different food sources for the bugs, thinking about maybe different regions of a field have different nutrients for the organisms. And what was really cool is we were able to identify a few subsets of microbes that seemed to support the growth of Herbispirillum, the symbiont, no matter what other nutrient variation or other strains of microbes were present. So they made the growth of Herbispirillum robust to other variation. And so this was sort of a toy example where we thought, OK, well, these subsets of microbes might be the kind of thing farmers would want to mix with a symbiont and like spray on their seeds before they plant them to help support the growth of crops. OK, turning back to a human health relevant application, tell you a little bit about how we deployed the platform in an infectious disease diagnostic context. And so this work started really before we got involved with the work of other groups at Broad who developed a really powerful CRISPR-based molecular diagnostic called Sherlock. And so this used CRISPR technology that very specifically identified DNA sequences, for example, DNA sequences coming from a viral pathogen. And so postdoc in my group, Sherry Ackerman, and got together with a postdoc in Pardis Sabeti's group, Cameron Mervold, and they put their heads together and they said, boy, isn't this Sherlock technology great? It can do a super good job of detecting one or a few target sequences or target viruses in a given patient sample. But wouldn't it be even better if we could use the combinatorial power of the droplet array system to test for many different types of viruses in many different patient samples all in one go? And that's exactly what they did. So what you're seeing here are some of the test results showing that a large panel of different viral detection assays worked across a large panel of test samples. And so what we had hoped to see here was that each viral test assay was specifically detecting the one test target that it was designed to react with. And for the most part, we got pretty good performance across this large panel, even with just kind of going through it a few times to optimize its function. And so that was really exciting. It was also exciting because we had started this work just a few years before the pandemic and had the paper in review when the COVID-19 pandemic began. And so we had, those of you with sharp eyes will see, we had a set of coronavirus probes in that panel. And with the outbreak, we added a SARS-CoV-2 specific assay and were able to include that in the publication, which came out very early in the pandemic. Okay, but with all the progress we've made managing that pandemic, it's important to remember that there are other really important infectious disease threats to humans. And bacterial pathogens being a particularly scary one, especially the drug-resistant, antibiotic-resistant bacterial pathogens that you've certainly read about. And so another student in the group, Gautam Thaku, decided to do the same kind of large scale assay where we could test for, in this case, many different bacterial pathogens in many different patient samples at once by adapting this assay strategy so it would work on larger bacterial genomes that had different characteristics than the viral genomes targeted in the earlier study. And so Gautam also streamlined the testing procedure to make it easier to use, and further than that, developed an alternative workflow where the person performing the assay didn't have to do any of the droplet microfluidics, any of the salad dressing stuff. All the diagnostic content was pre-populated in the microwell array such that the sample just had to be added. And then he even further showed that we could do the readout, which is done by taking pictures using a mobile phone camera instead of a research microscope, potentially making this a lot more deployable close to the point of care, like in a hospital or even in a low resource setting here in the US or abroad. With that, I'm going to hand it back to Alex to tell you more about how Broad Science is coming to patients.
Thanks, Paul. And very cool. And I just wanted to highlight some of the things at the end of what Paul had just said, which is that a lot of what we think about when we think about the Broad is we think about it as sort of this nexus where people from all around the Boston community come in to interact with one another. It brings together people from MIT, Harvard, the medical schools, and the surrounding hospitals to really do science that is difficult to imagine doing in other contexts. But inspired by a lot of the work that's been done, for example, by the DSP, sort of the data sciences platform here, we began to wonder if we could invert the model where instead of just doing this here, could we take these simple things anywhere? I mean, you all have now seen this little device, and you could imagine that this little 1x3 slide goes anywhere that you want. It does have this other little piece that we repurpose that comes with it, but it's pretty compact. And so what would it really take to invert this paradigm where the technologies go out into the world so that these sorts of things can be done anywhere to solve problems that impact all of humanity? Well, to try and test locally whether or not this sort of stuff was feasible, we paired up with Sarah Fortune and Brian Bryson over at the Reagan Institute. And Brian was a postdoc at the time. He's actually now a faculty member at MIT in Paul's department, who's doing fantastic. And we asked Brian if he would throw on one of these spacesuits, go into the BSL-3, this high biosafety level containment room, and run some tuberculosis samples and give us the thumbs up or not. He gave us sort of the thumbs up, but he generated this really beautiful data that showed us that when we looked at some cells that we thought were pretty uniform responding to tuberculosis, that actually we ended up getting 3 different response populations, and there was more complexity than we had appreciated before. But what this really told us was that these kinds of assays could be run under very complicated settings. And so you might say, well, this is run at a lab at MIT. Could you take it out somewhere else around the world to try and see how useful it would be? And so Mark and Travis here in the front paired up with Al and Henrik, who are in the back, to see if we could apply this to places where TB is a major problem and where there's a disproportionate amount of disease. And so we went down to Durban, South Africa, which is on the East Coast, and we tried to see if we could apply the same technology to real clinical isolates to see if we could make inroads into understanding what's happening there. So here's for truth in advertising what some of these tissues look like. They're pretty necrotic when you get biopsies, but you can see within them that, interestingly, there are regions where the immune system has actually been able to resolve the infection. Those are these healed scars that I'm pointing up at the top. And there are other ones where the bug is actually winning. And that's one of the most interesting things about TB, is that within the same person, you have these local battles where sometimes the body wins and sometimes the bug wins. And so Al, who unfortunately for him was on a game show, a Wipeout equivalent, and that's the picture I stole from it. I'd encourage you to go watch it because it's really funny. And Ian in his lab went and applied this, and we were able to generate exactly the same sorts of plots and data that we showed you before, coming from our labs either here or at the Reagan or at MIT. And so nice separations of different populations that I'm showing you in colors, and really clear gene expression patterns I'm showing you at the bottom, with each block of genes, those programs, representing one of the groups of cells I show you above. And so, what it turns out is that actually it's possible to take these technologies out anywhere. And so Mark and Travis decided that they would be the unofficial spokespeople of the lab and buy as many plane tickets as was humanly possible to go to as many places as they could. And so what we've really tried to focus on is how we can use these technologies to enable and empower others, going and setting it up on site and enabling them to do science. So, examples of doing some of the HIV TB work in Durban, and obviously no fun at all, as you can tell. And then, worked with Pardis and others down to try and figure out what's happening in Ebola, flights to Bangkok to try and get a better insight into what's happening in malaria within the liver, and even down to places in Latin America like Rio to try and understand what's happening in leprosy and why some people can resolve infection whereas others can't. And so, I don't want you to think that this is only applicable to infectious disease. We just happen to choose some examples. Another study that, that we did that I think was a really good example of how these things work is a study of environmental enteropathy, which is this disease that's associated with malnutrition and growth stunting that has to do with, issues with access to clean water and food. And so Travis and Connor went down to Zambia, where this is endemic, and basically tried to use this technology to understand what was happening in the gut that led to malnutrition. Because obviously, malnutrition stunting leads to major impact on local health. And so what people had seen previously is that if you look in the microscope, basically these villi in the gut, these little projections, these little finger-like things that absorb all your food, shrink down, and that is associated with increased pathogen defense. By taking small biopsies with partners in Lusaka, what Travis and Connor were able to find is that actually what's happening is really unusual. Some of the cells in your intestine are differentiating into a cell that's normally only found in your stomach. And so you get these cells that I'm showing you at the point right here that express this particular marker, DUOX2, and all these things that people had previously found as being associated with environmental enteropathy, we now find are associated with this specific cell type. And what's really happening is that this exposure is causing your gut to differentiate into your stomach so it can protect itself from pathogens. But in the process, it's basically blocking nutrient absorption. And so this gives you an idea of what is the process you would have to counteract to restore nutrition within these populations. And so these are a couple examples that I've given you, but we've actually worked with partners all around the world. At last count, we were working on 6 different continents with people in over 30 countries in hundreds of partnerships, really trying to set people up to run these assays by themselves for their own research. And through the HCA that I mentioned earlier, I've been deeply involved with a couple others in leading equity efforts, which the Broad has been very generous in supporting, as has the Reagan, to really try and make sure that these sorts of technologies can be applied to problems all around the world to help people wherever we can. And so I hope what I've sort of showed you today and what Paul showed you today is that there's really important things that happen when you bring together communities of people that come from diverse disciplines. If we can bring together deep biological insight on which problems to go after, which tissues to go after, bring together engineering know-how to sort of, MacGyver or however you want to think about it, solutions that let you go after these problems, and then bring together a little bit of the computational magic that I showed you before, it really brings us to a place where we can make transformative discoveries, not just here, but broadly. And so I want to thank, at least on my side, all of the people with whom I work. They're all fantastic. The best part about being in this community is the strength of the young scientists who are much smarter than I am and know much better than I do, which is what Paul was trying to say very gently. And I'm really excited to work with them every single day. I want to thank a bunch of our collaborators, and I want to thank primarily the people who participate in these studies who are our partners. And I don't mean the scientists, I mean the people who donate samples, because it's not lost on me that none of this happens, none of this global health work happens without people that are willing to be partners in the science. And I think we don't spend enough time thinking about what we owe them, but I spend a lot of time thinking about what we need to do to be responsible stewards of the trust they've put in us. And I'd like to thank our funders who've been very generous and hopefully will continue to be. And Paul's got some acknowledgements too.
That's fine. So really like to thank these folks. I think we've got folks folded who contributed directly to what I presented today, and also appreciative for funding sources. And with that, I think we'll take your questions.
I think they did a pretty good job. Thank you both. So if you have any questions, please, we encourage you to come down and invite you to come down to the microphones to ask. And we'll be checking the YouTube chat. My colleague Alex down here in the front has been monitoring the YouTube chat, and we'll be feeding questions over to here fairly shortly. I wanted to start off, though, by asking the both of you kind of how you got to be where you are today. And I don't just mean the Broad Institute, but as you have both come from through different paths to come to this place where you're both working at this intersection between biology and engineering, how did you do that? I think especially for any students who are watching today, they'd be curious about your career paths and how you got here. Now, don't be too polite.
All right. Well, let's see. Where to start? So I was trained as a scientist. I also have degrees in chemistry. The first one from the University of Washington in Seattle, so as a public university kid. I think, but obviously now, as Tom introduced, I'm a faculty member in an engineering department at MIT. So like, how did that happen, right? I think the important part of my story is that, although I was trained as a scientist, I think I'm an engineer in my heart in a lot of ways. And so, that fusion, was there for me all the way through my studies. And so, I think what's interesting about education, and this is true in my experience, and it's something I, try to support students I interact with on, is there's like all your teachers and at a university, all the faculty, teaching you stuff. So it's like, the student learning from the outside world. But there's also this process of self-discovery that happens at the same time. And so for me, a lot of what I was experiencing from the outside through the formal curriculum and research experiences and so on also helped me recognize things about myself and what appealed to me and what my talents were. And that was also really important to finding the right way to forge a career forward, where I could, leverage what I was able to contribute and help others along the way as well.
Yeah, so I didn't have it quite as figured out as Paul, but I will say that there's some important things I learned along the way about myself. I won't say that I have talents, but tried to do what I can. I'd say that I was probably like many of you, I don't know in this audience, but at least maybe some of the people that are watching online, where I really wasn't sure what I wanted to do with my life. I enjoyed science, I did okay in it, and when I was trying to figure out what I was going to do after college, I applied to everything in the universe, looked at a stack of different things, and fortunately my parents gave me a small nudge and said, you can go and do these other things later, why don't you go see if you like science? And so I went actually to graduate school in basic science, and I had gone to a liberal arts college because I wanted to study music, humanities, philosophy, everything in the universe, because I had been interested in everything. And so I went up to actually do pencil and paper theory work, where I was going to write equations on a piece of paper, which sounded great to me because I was told I could live anywhere in the universe as long as I had an internet connection. And so I had these dreams that my PhD was going to be on a beach, definitely not sipping a Mai Tai, but I was going to be relaxing, I was going to be enjoying myself. Went up to graduate school, not in engineering, but in science, and started talking to people, and I got matched up with this advisor who pushed me to tackle a very different problem, how the brain works, which obviously I was in a good position to tackle because at 23 I knew what I was doing. But in trying to figure out how to do it, it was how do we get this done? How do we move forward? And what I began to realize is that I'm really an engineer at heart. Like what I like to do is I like to see problems and try and devise solutions to them. Now, what my advice would be to anybody is like what I've done along the way, to Paul's point, is I've followed what is interesting to me. When I've had questions or things haven't made sense, I've gone after it. So I moved from being somebody that did pencil and paper theory to doing network neurobiology, trying to understand things like memory, learning, and logic, to doing immunology, to building devices that are like the ones that we showed you today that can be applied to study problems all around the world. What I've really come to appreciate is that what I like working with smart people on hard problems, doing things that I couldn't do alone. This is an incredible community with incredible depth and expertise. And what I came to appreciate was that there was this opportunity to work with all these different people to do things that I could have never imagined was possible. But even more than that, I began to wonder, why are we only doing this here? Why are we only taking advantage of people in Boston? And recognizing before the pandemic that everybody was just a plane flight away, it became really easy to go and work with people all around the world. And I think what I've learned, and social networks, all these kinds of things have made it clear, is that this is a small global community. It's a place where we all have the opportunity to interact with one another and to impact one another. And there's no reason we shouldn't do that in science. And it really enables things that you could have never imagined were possible. And it pushes you in ways that I find exciting, that keep me here and that keep me excited to come in every single day.
Well, I give you both kudos for starting in science and finding ways to stay within science. I started in science and ran away screaming as soon as I realized how bad I was at it. Do we have any questions from the audience? Yes, coming up. Let me just make sure this is on.
Hi there. My name's Ellen Simmons. I'm a biomedical engineer from Cambridge Consultants. We're like a product development firm, so we're often working in that intersection of different disciplines. I often find that one of the hardest aspects of working collaboratively across those, though, is that you're kind of speaking multiple languages, and so the biologist speaking to the engineer, speaking to the computer scientist, and sometimes that could be quite frustrating, that could be quite difficult. I just wondered if you could speak to that, maybe ways you're addressing that as a difficulty or some examples of where you've overcome it.
I think you've hit the nail on the head in terms of the hardest thing in interdisciplinary science. It's that every field has its own language. Everybody has their own way of talking about the same problems. And one way of looking at it is it's a problem. The other way is that to say, well, it gives you an opportunity to see the same problem from multiple different vantage points. But what you need to do is you need to have a common language for discussion. So there's no simple way of doing that. Usually what it takes is bringing people together, creating communities, creating places and spaces where people can talk and learn languages and go back and forth. making sure that there is that strong sense of community, that people are willing to be vulnerable and are interested in one another. And I think that that's what's so great about places like the Broad. You know, they really bring people together and create that opportunity for dialogue that wouldn't happen in other places. I know originally what drew me in and has kept me here was a random side comment where somebody said, this technology you're developing, which was this really weird like bed of nails that could be shoved into cells to deliver molecules. They said, well, is that applicable to other stuff? Why don't you come talk to this community, hear about all the problems that people are having, find a place. And you know, that decision to help somebody because so many people had helped me snowballed into lots of different projects and eventually into trying to figure out why those cells weren't doing the same thing, which is what led me to some of the stuff I'm talking about today. So there's no simple way to do it other than to bring people together and to have sort of a place where people are willing to be vulnerable, not interested in showing how smart they are, not interested in showing how accomplished they are, but really just willing to try and learn from one another. And you know, I know that students tell me all the time about how nervous they are, like how they're going to go into their committee meetings and how they're going to find out that they don't know something or it's not going to go well. And I say, well, you know, the only thing I learn every single day I'm here is how little I know and how much there is to know. And so, you know, don't worry, I feel even more like a fraud the more I go forward. And so I think it's curiosity. I think curiosity and engagement. And that's what I like about this community, but there's no reason it just has to be here. It can be all around the world. Sorry, Paul, you're up.
I don't know how much I can add to that. I think there are generational changes too. And so, as I'm sure you're aware, there are a lot of students receiving their training on a multilingual basis now, who I think will be able to enter organizations like yours and have a really big impact at the interface between those different groups of professionals. Yeah.
Thank you so much.
I think we have another question from the audience.
Hi, thank you both for speaking. That was a fantastic talk. I have a technical question about the pico wells and the micro droplets. Like, how do you actually load the wells? Like, what's the frequency of wells being empty or doubling up on cells? And then when you do the like multiplexed like compound delivery, are you able to control like how many droplets containing which molecules actually make it into the wells that you care about?
You want to start with?
I can start with the pico well part. So. The answer is that we don't actually try and be too deterministic. So we have 100,000 little wells, and if you actually drop 5,000 or 10,000 cells on top, the probability that any well gets more than one cell is low. If you want to reduce it down lower, you load 100 cells, you load 200 cells, but if you load 100, you don't get 1,000 out. And so we actually went through and we figured out for every single loading, how many doubles would we get on average? And we imaged it under a microscope. And it's probably like that probability problem that you hated in high school math, where they asked you if you drop a pin on a piece of paper, what's the probability that it crosses some lines that are ruled? I still can't solve that one. But I can tell you, at least for dropping the cells in the wells, that it's like under 1 to 2%. And if you want to go lower, you can. There are tricks where you can actually load together cells that have different tags on them or that come from different people, and you can actually load many in a well and still figure out what belongs to whom because of differences in genetics or differences in tags. And then I'll let Paul explain how it works more broadly.
Well, we're using the same math, and one way I kind of think about it is we have a brute force solution to the problem, so we're not looking to beat the statistics. It's all random loading. In our current implementations, we just make the arrays really big with lots of microwells. So even if it's just 25% of the droplet combinations that are of interest in our study, 25% is a lot. And then, of course, someday there will be a student who really gets excited about beating the statistics and using some of the strategies that you're probably thinking about, like different droplet sizes and asymmetric well designs, to get that last 75%.
But simple is great.
Alex, I have a question for you, and then I'm going to turn to other Alex to see if there are any questions on. Oh, and we have somebody else on stage as well, but let me get my question in first. I'm fascinated by this idea of minimizing the technology and being able to use it in as many situations and places as possible. Mm-hmm. But you're minimizing the actual amount of material you need in the slides, but what about the other equipment? Is there a point in which you're going to be able to throw whatever you need into a backpack, take it out into the field with a battery and a laptop, and use it in any place?
So it's a great question, and I'm going to be very careful about how I answer this because it's going to be on the internet and my students are going to hold this against me. So I will tell you that when we started off doing this, that my students would load these things in their underwear. They'd wrap them up in like their suitcase and bring them with them. I didn't need to be in there, but I think they wanted to tell the story to their children someday that they had thrown these devices in their underwear. Now, you can imagine you've seen these little chips. They're small. When you pack them this way, you can get a large stack of them. We have these little clamps, which we didn't make. We repurposed them. They were expensive. They were lying around. And basically, we use this to put this little device in and seal it. Now, it turns out you need one other thing, which is a little bit of heat. So you can do this. You can basically take it anywhere, and you don't need anything fancy. We've done it with these fancy clamps. We've done it with a T-shirt press. We've done it with a George Foreman grill. And one of our, you shouldn't eat on any of them afterwards. And one of my technicians at the time went into a cave to look at bacteria and decided that the best way of sealing it with a little bit of heat was to put it underneath her armpit. I did not know about it at the time. I do not condone that. I do not recommend it. But hypothetically, all you need is that little chip. But please don't do that. Please bring a clamp or at least buy a portable grill.
That is not an advertisement. We do not endorse or condone the use of George Foreman grills for scientific experiments. Question from the audience.
Yeah, thank you. Great talk, both of you. It's always inspirational to hear what you're up to and feel like you're seeing the future. But I'm wondering, the endpoint that both of you described was really publication or kind of proof of concept work at small scale, in different settings. And I'm wondering, what is the endpoint and what is the vision, and how do you get from here to real world implementation at scale? Or is that even what you're trying to do? Are you kind of presenting the proof of concept and letting others take it forward, or where do you want to go from here?
Well, I'll take that first. This comes back to our very first slide, our disclosures slide.
Oh, I missed the first slide.
Yeah. That's okay. There's a lot more to say about it. So I think, speaking for myself, the goal is absolutely out beyond that publication. We want to have the impact out in the real world that affects everybody. And so the question is how to do it, because it's not easy. It takes, and a lot of times people talk about bringing in private investment and that kind of a dollar resource to support the translation and commercial development. That's true, but it takes a lot of expertise on top of that, and expertise that's different than what I have. And so what I've found is it takes a concerted effort to find those partners and recruit them to make a plan to try to figure out how to let the technology take flight and have the kind of impact that you're talking about. And so that's one of the reasons why I have engaged in commercial translation beyond my work at the Broad Institute. And that's a big part of the endeavor as well. And I'm lucky to have some great partners in doing that.
So I'll just add to that. I'll say that, for me, the goal is to help people. What gets me excited is helping people. And, what Paul said at the beginning about what we learn about ourselves, what I've learned is that I like helping people. But I recognize that in academia that I have different goals. My goal as a faculty member at MIT and at the Broad and the Reagan is to mentor students and to enable and empower them to pursue the things that they care about, to launch their careers. And so I have to think about things like publications and how to support those things. And I recognize that there are systems within which they need to operate. Now, on the other hand, I personally think it would be criminal for these things not to be getting out broadly, for the technologies, for the insights. And that's where partnering with people in sort of surrounding biotech and in large pharma becomes so important. And to me, it's part of that constant learning. You're learning new processes, new operations, and part of going out into the world is understanding where technologies don't work, thinking about how you can innovate further, figuring out how you can make them better, figuring out what are the things you have to do to impact local problems for people around the world. And so I think it's a different set of skills. I'm only beginning to learn it, but the rest of my family's happy.
Especially in low-income settings and resource,
It's some of the best things I've ever done, and it's some of the places where I felt like it was the greatest and most significant work I've ever done, because I could see the relevance. And when I see people using these technologies in their own labs to publish stuff during the pandemic, out of South Africa or other places, like that's really what excites me. But I think my family's excited about it too because I'm the only one who does science. Everybody else is sort of on the business side of things, so they no longer have to listen to me talk about science. They can listen to me talk about the business of science, and at least they feel like we can have a conversation.
Awesome, thank you.
Turning to the internet, Alex, do we have any questions coming in over YouTube?
Yeah, this one's from Kritika Online. What percentage of engineering approaches to understanding biology still involve the use of scientific methods of hypothesis generation and testing?
So I'd say all of them. And I think it's one of those things where we always have a hypothesis. We just have to be very clear about what the hypothesis is. People say genomics is fishing, and yeah, I mean, genomics is you measure everything, but at the end of the day, there's a hypothesis. It's that if I compare this sample to this sample and measure everything, that I'll be able to find differences that are meaningful. So we always go with the hypothesis. We have an idea why we're trying to do something. We just have to be very clear about articulating that ahead of time, getting back to Paul's points about stats. In terms of making sure we're powered, we have to make sure that we are not, rewriting it later on. And if we don't think about what we're really going to test, we don't structure the experiments properly. And so I hit this in that last slide where I said it's biology plus engineering plus computation. I like to think that we think about problems, we think about engineering solutions. We also think about sort of the broad applicability of those solutions. Is it a one-off? How big is the problem? How important is it to solve it? But at the end of the day, there's always a hypothesis about if we miniaturize, we'll do this, or if we simplify, we'll do this, or if we compare this with this, we'll find this, which we can then go test. And so I think at the end of the day, it's important to frame the hypothesis part of it. It's just that we can be broader. And so rather than just saying, let's focus on this one protein, we don't have to make assumptions. And the biggest thing that scares me in all the beautiful images that I see on screens is always what I'm not measuring. If I see a picture of a neuron and it looks very elaborate and it's got dendrites going everywhere and it looks like this tree, I'm always what are all the other things that are around it? What is it talking to, and what am I missing? And so we have hypotheses. We just have the opportunity to look at them at this unbelievably broad vantage that I think really gives us the opportunity to find things that matter, as opposed to trying to look under the lamppost that we have.
I love this question because it goes right to the heart of what this series is about, discovery. I agree with everything you say about hypotheses. We always try to make the best hypotheses we can and have those lead the science. There are also cases where discoveries crop up that are just outside our imagination, and we're outside the scope of where the sandbox in which we were thinking about defining hypotheses. And so one of the things that's exciting about the new technologies that we talked about tonight is it's possible to have it both ways. You can design an experiment that's grounded in a rigorous hypothesis testing, but also have the potential through the scope of the data and the magnitude of computation and so on to also make serendipitous discoveries that actually expand our universe of thought and enable us to dream up new hypotheses that are even more fantastic.
Anything else? No? All right. Well, I know I'm probably the last thing standing between everybody in the room and the reception. So with that, thank you both very much for a fantastic talk and a fantastic kickoff to the Broad Discovery series. So we hope all of you have enjoyed the talk tonight. All of you who are in the room, please join us in the lobby for a reception. A recording of tonight's talk will be posted on our YouTube channel. Look for it over the next couple of days at youtube.com/@Broad Institute. You can find other recordings from other Broad talks there as well. And if you're interested in keeping up with us, please subscribe and ask your family and friends to do as well. Thank you very much and have a great night.
The Broad Discovery Center is a public educational space that showcases the groundbreaking biomedical research happening at the Broad Institute, our partner institutions, and our collaborator sites worldwide. Through our interactive and informative exhibits, you'll learn some of the stories behind how researchers at institutions in Kendall Square and across the globe are working together to better understand and treat all different kinds of disease. Building the Broad Discovery Center has truly been a community effort. Our team of scientists, administrators, consultants, community members, and educators have all come together to build a special place for anyone to engage with cutting-edge biomedical science. Whether you have a background in science or not, all are welcome at the Broad Discovery Center. If you're looking to discover more about Boston-based innovation, we encourage you to stop by. Our interactive learning exhibits offer a deeper look at the complex biology associated with cancer, psychiatric research, infectious disease, rare disease, and other conditions. You can also walk through exhibits to learn more about the journeys, stories, and motivations that drive Broad scientists to pursue disease research. We are so excited to bring this space to you, and we hope to see you at the Broad Discovery Center very soon.